Our final speaker is Dr. Ryan Dilger from the University of Illinois. He's a professor there and working in the interaction of nutrition, immunology, and neuroscience. And he's going to talk to us some more about the mechanistic insights into soy-derived bioactive isoflavones, saponins, and modulating the immunity and improving disease resilience in swine.
Great, thank you, Paul. Thank you, everybody, for hanging around. I don't know if it's enviable or not. I'm a nutritionist. I'm not a DVM. We're going to talk a little bit about mechanisms here today, but thank you for sticking around. I want to expand upon what Brooke has talked about, hopefully give you a couple of insights here on the mechanisms while keeping it light, just giving you things to consider as part of this. So we already know the concept of where respiratory disease in pigs is causing an issue. My lab is interested in that nutrition by health interaction as part of this. And so in particular, we've been focused on the soy side and recognize all the wonderful support that US Soy has provided over the years. But I would say this is a story of serendipity that Dean had mentioned. I was simply in the right place at the right time and had some great mentors and great students, and Brooke included as part of this. She presented kind of the long-term penultimate part of her dissertation. I'm going to give you a little bit more broad view of this, but we really focus in my lab on this window of susceptibility for these young pigs. Right when they are about to be weaned, we're changing their environment, we're changing their diet at the same time, we're starting to bring soy into that. We worry about gut integrity, inflammation stemming from that, and any type of respiratory illness that's going to come thereafter. So certainly PERS is part of this, where we're trying to focus on that management aspect and use nutrition in this way as a tool to help that pig along. And so as part of that, as a nutritionist, I think about it from a metabolic perspective. And everything that we are doing is providing these nutrients to this animal to meet their genetic potential for them to grow. Largely that is lean tissue accretion. Those amino acids are very expensive, which is why I originally became a nutritionist, to get in this concept of 60 to 70% of the cost of raising that pig is in the diet. So as a nutritionist, I'm providing something that they absolutely need all day, right? They're eating multiple times a day. But as soon as that pig gets sick, their priority for nutrients changes dramatically. They stop depositing into muscle. They start to use those amino acids to build other proteins in the body that are necessary for them to get back to a healthy condition. Now, one challenge being a nutritionist is all of a sudden that pig is no longer eating as well as they were before. So the technology that I can provide, we're having to reprioritize that. But I think I want to help you understand where we can use nutrition to be a proactive piece of this. So how can we set them up for success as part of this? Ultimately, that pig restoring homeostasis is going to be critical. Obviously, if they perish, they're not eating anything at that point. But as part of this, we want to get there as quick as possible. So we think about the metabolic costs of activating the immune system. Again, we know there's both the innate immunity that's happening in minutes to hours after that pig has become infected, and then adaptive immunity that's happening days to weeks thereafter. Each of those has a metabolic cost, and largely here I'm thinking about it from an amino acid perspective, as that's a very expensive nutrient. But upon challenge here, we have this large response with the innate immunity. That's where the body is mounting that initial, largely pro-inflammatory response, producing a lot of inflammatory cytokines and other proteins, and therefore diverting those away from growth. And we start with that highly inflammatory side. It has a larger magnitude, shorter duration, and then we get into the adaptive response where we're producing those antibodies and hopefully having some memory for that pig seeing that same disease in the future. Now, each of these is costly to the pig, and we seek to reduce the frequency that pig actually gets sick, but especially the duration and the magnitude of those responses. So if we can shave a half a day off of that pig being sick, those nutrients then are going to go back towards lean tissue accretion as part of that. And largely we focus on intestinal immunity here, given that up to 70% of all immune cells are going to be at that interface. So again, where is the diet influencing immunity? It starts here, and where we want to be able to get them back to eating and back to a healthy state as quick as possible. Now, nutrition as a tool, again, is one thing that we can use to be proactive. If they are not eating, they're not receiving any of the nutrients, the technology that we're providing, but it's also a very practical way to provide this to the animal. So we consider this to be a very scalable approach. And by and large, I would say that using soy in this context is insurance. I'll talk about why it's a good protein source, But also here, there are other components of that diet that Brooke alluded to in these bioactive compounds, that even though they're at very small concentrations, they have a very large effect that they can have. We call these secondary metabolites that are emanating from that soy plant. So again, if we can leverage what's already in their diet, with soybean being present there, and be able to use that to help shape the immune response, proactively perhaps, but also during and even after in the recovery phase, this can have many advantages from a nutrition perspective. So our goal is that integrated resilience in terms of bringing that animal back to a healthy state. How do we use nutrition, again, to be proactive and pair this with good biosecurity and other pharmaceutical approaches as part of this? All right, so perhaps you've seen this graphic before about what is the soybean, how does it come together? But soy is already the cornerstone of swine diets in the United States. We are really pairing this with corn, and they have complementary amino acid profiles, but that soybean provides a high concentration of a good quality protein that we can be using for that young pig. Now I'm going to focus mostly here on these other smaller components called bioactives to particularly influence immunity. And these are going to be isoflavones and saponins, so going a little beyond what Brooke had talked about. And if you look at what's in an average soybean, you've got about 20% oil. The rest of that is going to be meal. And in fact, these secondary metabolites that we're talking about don't even make this list. They are a very small portion of the overall weight of that soybean. Typically in the swine industry, 15 to 35% would kind of be the range of soybean meal that's going to be provided in that animal's diet already. And I'll say that these are sufficient concentrations of that product to elicit the effects that Brooke was talking about. So already we have soy at levels where these biological active components can have their effect. Now, processing is everything here. We've got the raw soybean seed at the beginning. That's going to go through that dehulling and oil extraction procedure prior to being toasted. Once we do that, that's how we're producing the soybean meal. And we can go further to remove the soluble carbohydrate fraction to get to soy protein concentrate, or SPC, and further even still to get the insoluble carbohydrate removed to get to soy protein isolate. So soybean meal would be probably the most typical soy product included. We get to more specialty products as we're starting to remove some of these anti-nutritional factors. We've done a literature review to understand fitting into this concept. This goes back to the 1970s. So anti-nutritional factors here in blue. Largely, we've been processing soybeans in order to remove these factors that are negative for that young pig. And in doing so, it's focused predominantly on trypsin inhibitors. That's why we're toasting that soybean meal. And also a few others that fit into that. Carbohydrates would be the second most studied aspect of this, down through, and you have saponins and phytosterols here at the end. There's been very little research done specifically on these secondary metabolites in that soybean. But, again, as Brooke mentioned, we can use the fact that we have processing methods already that are producing these kind of value-add soy products because it also changes the concentration of these secondary metabolites. So it gives us a very unique opportunity to be able to start a diet that it basically is a purified protein source and then add other components that came from that soybean. So in essence, we take the bean apart, and the studies that we have been doing is to put that bean back together in a very particular way such that we can isolate the effects away from the amino acids and towards these secondary bioactives. So this is where we're going to just focus on isoflavones and saponins. I'm going to give you one slide on each of these so you understand where they're coming from. You will not have to remember the structure of these in any way, but these are very large products that are produced by that soybean. And by and large, both of these are defense mechanisms. The isoflavones and the saponins are defense mechanisms because that soybean plant can't get up and run away. With isoflavones, these are antiviral, antifungal for the plant, and antibacterial as well. But as Brooke had mentioned, there's three main types, the genistein, daidzein, and the glycetine. And as part of this, about 50% of the total is going to be in genistein and about 40% is daidzein. And I'm highlighting here the carbons 5 and 6. This is predominantly where they differ in their structure from one form to another. But overall, these structures are very similar. This large phenylpropanoid-derived compound with estrogen-like activity, if we want to get technical about it. But again, produced in concentrations that are higher in soybean meal, lowest in the soy protein concentrate, and kind of intermediary with the soy protein isolate. Now in each of those three cases, though, if we show, the proportions of genistein to daidzein to glycetine stays about the same. So relatively, all we're doing is changing the concentration, but relative to each other, they're staying in about this 50-40-10 ratio. Now as part of this, and my slides are a little bit messed up here, but as part of this, there's another one out there called equal. Equal is actually produced after microbial fermentation or conversion within the intestine. And equal is much more potent than the other three isoflavones as part of this. So if you read the literature, you'll identify that from a bioactivity perspective, equal is much more active than genistein or daidzein. And the literature is fraught with a lot of rodent research that says that equal is everything. We should be focusing on that one piece. I think my arrows are gone here. But if we look at what's happening with equal, in mice, it works really well. And basically, mice and non-human primates have a microbiota that can convert daidzein into equal. Pigs and humans do not. Less than 1/3 of all humans, when they've done a survey, and very few pigs as well, have that ability. So largely, we are focused on the non-microbially derived, what's in the soybean to begin with, for this activity, where again, genistein and daidzein are those which are most active as part of this. So one thing that's unique about the isoflavones, they can be absorbed, they will circulate, we can find them in concentration within the bloodstream, and as part of that, they can then have direct effects after being absorbed. So this is a dietary component, absorbed in the intestine, circulating to the lungs, can have a direct effect there in the lungs when it comes to an immune response. This differs when we talk about saponins. Saponins are also secondary metabolites. They are also meant to prevent or protect that soybean plant. And this way, saponins are very bitter. So this is also the antifungal, antimicrobial, but it's also keeping herbivores from eating them. So it can be very bitter. These are terpenoid or steroid glycosides with an amphiphilic property. So what this means is that these are large carbon structures that have some polar heads and polar groups on them. And because of that, they are soap-like. They are able to actually dissolve things, and this is how they become actually antiparasitic. We use these on the poultry nutrition world as well. Saponins derived from other sources are able to directly kill parasites in this regard. So two big groups, Group A, Group B, they really only differ by where they have carbohydrates that can be attached to these. As part of that, then, what's termed a sapogenol, this is where you have one of these two forms with carbohydrates attached to them, and again, microbial conversion is required for that to occur. So you get a lot of diversity if you're type A versus type B, and then which carbohydrates are linked at which particular positions. But here again, showing our three fractions in soybean meal, the soy protein concentrate and the isolate. Soybean meal, again, has about 3 times the concentration of saponins that it does isoflavones. So both of these are present, and in this way, it's mostly all the group B saponins that are present here. Now, different than isoflavones, these saponins do not leave the lumen of the intestine. They cannot be absorbed. Therefore, they're not likely to have any direct effect on immunity. They're going to have indirect effects. But gut immunity is going to be a big part of this as well, and they will act locally in that regard, and I'll elaborate on that. All right, 2 lists, mechanisms of action, some overlapping between saponins and isoflavones. Largely, they are anti-inflammatory, and that's where we start. Second, they have antioxidant defenses, and we cannot separate inflammation and oxidative stress as part of these. Both are important. I think it's almost impossible to be able to separate one from the other. But in this way, both of these categories of soy secondary bioactives are going to have positive effects here. Specifically, isoflavones also support the adaptive immunity and really the T cell development as part of that. And they have the direct antiviral activity that Brooke alluded to as well. And very much understudied is this concept of what they're doing in binding to the estrogen receptor, what that means from an immune perspective as well. A little bit different on the saponin side. Again, these are working at the level of the gut, stimulating epithelial barrier function, and making sure that we reduce the ability for secondary bacterial infections. So with that PRRS infection, it's opening the door for there to be a leaky gut, if you will. Saponins can help to make sure that that does not occur. And they do so partly by stimulating mucosal IgA responses and production there. And again, mentioning antiprotozoal activity, not as much of an issue in pigs as it is in chickens, but that's a unique feature of the saponins as well. All right, I'm going to try to keep mechanisms light. There's lots of arrows going on here. But in this concept, one of the ways that both of these secondary metabolites from soy are working is to stimulate and regulate this master regulator of inflammation, NF-kappa B, nuclear factor kappa B, and another one called STAT1. So NF-kappa B sits here. We can link this to everything from T-cell differentiation here with macrophages in pro-inflammatory cytokine production and otherwise. It is largely shaping the way that that pig is responding to any type of infection, and again, helping to set the stage for both that innate and adaptive immune response that are to follow. So if there's one factor that's going to be important, it's this in NF-kappa B, and it's tied to everything, including the oxidative stress that we'll talk about in a little bit. But here, both isoflavones and saponins are known to inhibit the activity of NF-kappa B and therefore help to reduce the inflammation that's going to be produced. So less TNF-alpha, tumor necrosis factor alpha, and interleukin-6 as primary pro-inflammatory cytokines. It's reducing those by preventing the translocation. It's also limiting overall tissue damage as part of this. So again, partly what's occurring when that pig is going through a respiratory disease is they are starting to use their own body stores, largely that protein that's found in muscle, to release amino acids to produce all the cytokines and other inflammatory markers that we have here. They are not consuming more protein, therefore they're using their own body stores to do so. Survival is key. They're repartitioning those nutrients, and that's going against what we're trying to do in allowing these pigs to grow. So there are multiple avenues where these bioactives can support immunity as part of this. Again, this is just a broad level part of where NF-kappa B is coming in, but largely they're doing that by taking these extracellular signals and translating it and therefore converging this biological mechanism. So this is an aggregating event. What we mean by that is any immune cell is going to have a receptor on its surface that it can recognize a cytokine, or it can recognize we've had a bacterial infection through a toll-like receptor. So this is how these immune cells actually detect what is happening in their environment. They are detecting that there is E. coli and they need to have a response. They're detecting that there is inflammation and we need to make more of that inflammation. So in aggregating here, all of these receptors come through this process. NF-kappa B is found here in the cytosol. NF-kappa B is kept in that location until it needs to be called up. And as part of that, it is held by I-kappa B. And basically, any of these extracellular signals are allowing this kinase to phosphorylate that. It dissociates, and that allows NF-kappa B to then translocate into the nucleus, bind to its nuclear receptor, and increase the production of lots of different types of genes. So if any type of inflammation is occurring, it's going to amplify that signal over and over and over again. That signal may be that we've had a weaning stress. We've had some type of damage to the gut or otherwise. It could be for any reason, but inflammation begets more inflammation, and it goes very quickly. And that's why that adaptive or that innate immune response went so high so quickly and was using a lot of those resources. Very similar when it comes to interferon alpha and gamma here, extracellular. Now here, STAT1 and 2 are able to translocate in and do their signal as well. And as part of this, acknowledging that we're talking about mouse macrophage lines here, just found some evidence from 2007 looking at providing daidzein and genistein directly onto these macrophage cells, which have been stimulated through LPS, so think through TLR4, the ability for that cell to respond to its environment. And with the presence of daidzein and genistein, basically there was less P65, which is the NF-kappa B, showing that there's a direct effect of isoflavones on the ability for that master regulator to do its job. So by taking away this signal here in the middle, we have significantly reduced the amount of inflammation that's going to occur, and the same happened with reduction in the translocation of STAT1 as well. So there hasn't been anything that I've found in pigs directly looking at their macrophages as part of this, and this is only one part of the inflammatory response, but it is promising for this being one of the ways that soy isoflavones may be working as well. On the other side here, we have antioxidant defenses. And again, this is a piece where we know that oxidative stress is going to be an issue, and we cannot separate that oxidative stress from the inflammatory side. Both are happening and they're feeding into each other as part of this. So largely, we have to have a balance of antioxidants in the ability to stop any oxidative stress. As part of this, we have the production of reactive oxygen species, or ROS, from many different sources. And when it comes to a respiratory illness, that's intentional. Many of the immune cells we have as part of the innate immunity are producing reactive oxygen species in order to kill the pathogen that is present. So that is absolutely intentional, but it can get out of control. And when we have more pro-oxidants than antioxidants in that balance, we get what's called oxidative stress. And as part of that, we can lead to mitochondrial dysfunction. We can lead to tissue damage. So all these coming together, again, inflammation begets oxidative stress, and they go into a vicious cycle. There are mechanisms to help reverse that, and that's where, again, where nutrition can come in to be able to help quell any of that inflammation and oxidative stress over time. So just one example here for alveolar macrophages. These mitochondria are producing reactive oxygen species. This is the electron transport chain. Eventually, you get leaked electrons and the production of superoxide. That means that it has an unpaired electron on it, and that's what's actually causing that damage. For some immune cells, this is intentional in order to kill the bacterium that's coming into play in contact with, but we call this a phagocytic respiratory burst. The problem is when we let oxidative stress get out of control. We don't have any mechanisms there. So largely, we try to have antioxidants in that animal's diet. We try to help them produce the cure to what they're going to experience. But as part of that, both isoflavones and saponins help to support the antioxidant piece of this. So helping to reduce that over time. Now finally, and mainly on the saponin side of this, for mucosal defenses happening at the level of the gut, again, intestinal integrity has to do with these tight junctions between individual epithelial cells. So the intestine is a large surface area where we are constantly into contact with antigens coming from the diet, something that could stimulate immunity in this case. So it's the outside world in the lumen of that intestine versus the inside world where things have been properly absorbed. The issue is that if we have a breach in the tight junction proteins, we have a leaky gut. And again, this is where that weanling pig is especially prone to these issues in terms of inflammation and other maladies, because this means we're going to get secondary bacterial infections. So as part of this, saponins in particular are helping to create a better barrier here, make sure that we have those tight junctions working, we have proteins that are being produced and creating a mucus layer such that the resident microbiota that's almost in direct contact with those epithelial cells is not able to actually translocate into the bloodstream. So overall, saponins can support immune response via the gut-lung axis, but it's probably not direct. It's going to be indirect there for preventing any type of secondary infection that may occur. All right, so just to dial back a little bit in this progression, I talked about this being serendipity, and I love the way that Dean laid this out in terms of seeing something in practice, which we wanted to understand better to bring into the laboratory with a very controlled setting. But the original work by Laura Greiner at Iowa State back in 2001 really started before we even made these observations in the field, and started with looking at individual isoflavones. We then went on to the study that Dean mentioned, with the benefits here for feeding high soy diets. And I'm going to just talk a little bit about our original work on that, where we just confirmed that piece, but in the same vein also isolated the fact to not be amino acids and focus predominantly on the secondary metabolites in isoflavones and saponins. And then one of Brooke's earlier studies here talk a little bit about the T-cell response in these PRRS-infected pigs that set the stage in the weanling pig for the longer-term study that she had already talked about. And then again, just confirming from some of her work, you did so much work on this, that the microbiota was not involved in this, right? Trying to rule out some of the different factors that were at play as part of this. And we have a number of different literature reviews out there as well. If you'd like to read more on this, how do we get to this place? What do these bioactives mean? So lots of literature here over the last 10 or 15 years we've been working on. So just one slide for each of these two studies. But first, again, confirming as we brought this into a controlled setting and out of a field situation, that increasing soybean meal from 17.5% to 29%, again, isolating the effects of these isoflavones, meant that we could rule out this being an amino acid effect. So here, in simply changing the amount of soybean meal that was in those diets, we had a 20% increase in average daily gain. The second week after that, and the full 2 weeks after infection with PRRS, and we significantly reduced the viral load as well, measured by PCR at day 14. So again, can we reduce the duration and the magnitude of that immune response following this respiratory infection? Partly, we could look at inflammatory mediators like haptoglobin, which at 3 days was about 40% reduced in these pigs that received higher soybean meal diets, and even in TNF-alpha at 14 days. So again, reducing that duration and magnitude of inflammatory response is part of that. So this was just being able to prove to ourselves that what we saw in the field could be replicated in a very carefully controlled setting. And then as part of Brooke's work again, then isolating the effect of isoflavones. And I will say we're being cautious. We say isoflavones, but if you notice the product that we used there was 40% isoflavones. The other 60% was likely saponins. We don't know that for sure, but we believe that to be the case. So we kind of talk about these now together. So in this case, we're feeding at 1,600 milligrams per kilogram with the soy isoflavone enriched product. And just showing here at 3, 6, and 14 days, immediately we see a reduction in the amount of interferon gamma. So we're immediately reducing that inflammation up front, which in itself sounds a lot like what PRRS is doing to evade detection by the immune response. But it's about the profile of all the cytokines being produced for shaping that ultimate T-cell response. So we can see this. This is the percent T cells that are isolated from peripheral mononuclear cells. And as part of this, we have a significant increase in those pigs that were supplemented with isoflavones, and also changing that ratio of helper cells to cytotoxic cells throughout that process. So again, shaping that immune response through the diet that they were consuming before and somewhat during, though they obviously don't eat as much when they are infected with PRRS. And ultimately then skewing the response of these T helper cells. Again, this is skewing more towards the Th1 side of things, which is the positive way to go, and less against the IL-4 mediated piece in these pigs. But we are shaping that immune response all because of the diet that they were consuming before they were infected with PRRS. So this gets a little bit tricky, given that IL-4 doesn't work the same way in pigs as it does in mice. And largely, we're probably now chasing the story of, is this a Th17, T regulatory cell? There are different ways that this could be occurring. But it all begins with these isoflavones being present in helping to shape that immune response. All right, so right now what we are doing is trying to even separate further the isoflavones from the saponins. So in my lab, we're also working with a chicken model where we're using coccidiosis and LPS to understand innate and parasitic infection as occurring here. So trying to isolate these effects and identify where we think more of these are going to be occurring, but really focusing on proactive and practical ways that we can use soy in the diet of these animals for being able to support that innate and adaptive immunity. And it's interesting talking to colleagues at my university who say, well, what would you like to increase or decrease as the primary soy breeders? And they say, not a problem. If you want more saponins, we can certainly do that. So it seems like the opportunity is there with genetic manipulation and otherwise to really dial this in for kind of soybeans with value added as part of them. And some of the take-home messages here from a veterinary clinical implication, We are already feeding these products. I think we need to give more emphasis to what they are doing. Right now, we are simply looking at soybean meal as being risky for that young pig, but we know a lot about how to feed that soy product early in that growth cycle. We know that isoflavones and saponins are both naturally occurring and complementary in how they are working. So as part of that, using these to our advantage. We know that this is not a replacement for vaccines or therapeutics, but it is complementary and should be taken as such for helping that pig along early in its life. And finally, having some clear and practical dietary strategies to help to support this immune resiliency as part of this response. So I know that was pretty quick, but I just want to acknowledge some of our key collaborators here. Dr. Mark Burhow, who's now retired from the USDA ARS, has helped tremendously in the analytical side and enabling us guiding us through this work. Graduate students past and present who are still working on these projects, again, trying to understand these components and a lot of work that goes into doing these biosafety level 2 types of studies. Again, acknowledging all the support from the US Soy Board and also our industry partners who helped all make this possible. So very quick, left you with those different pieces, but we are happy to answer some questions here today. So thank you.